back up.
There's a hermit crab on the sediment too. Poking around there.
Yeah. Yeah, it's interesting how the sediment on this, where we have seen sediment on this seamount, which hasn't been in a lot of places, as opposed to other seamounts that we visited that had really like a covering of uniform manganese nodules in the sediment patches. Here it's much more patchy and sparse, but there also seem to be a bit larger. You said we have space in a box?
Yes, yeah, we have starboard and forward box space.
Okay.
Are we pausing here?
Yeah, if we could, just for a brief moment. Looks like there might also be some xenophyrophore in there.
Yeah, there might be some infauna in here.
Yeah.
Seeing some structures that would suggest worms.
Some healthy sediment. Yeah, so
please.
Jess, if we could try to get a scoop in here.
I think this is the scoop's been previously used, correct?
I do not know.
Scoop has not been used.
I hear it's in the forward bin.
Oh, it might have been replaced, but yeah, I don't have.
All right.
I don't have any samples in the forward bin, so it may have just been placed there.
Roger. Between a rock and a hard place where this is sitting down. Any of these parking spots. So do you guys just want the rocks, or do you want the sediment as well?
The sediment is less of a priority. I mean, we're going to get some anyway, but more on the rocks, please. And trying to get a variety of different sizes.
Roger. Jake, you want to open up that box there, the toolbox, please?
And can we get a closer zoom of the rocks right in front of us before we scoop them?
Yeah, sure thing. Go ahead and push that in.
Looks like at least one of them's got a tiny little
There's actually a carnivorous sponge on the lower center.
That
on
one right
that
there?
nodule. No, right center screen now. Very like feathery looking structure.
This one right here?
Yeah, it's
Okay.
a sponge, carnivorous sponge.
Oh, is it? Okay.
Asbestopluma, yep.
Yeah, so let's try to scoop in that general region.
Oh, I please.
Get some of those different sized nodules.
Jake, you want to open up the toolbox, please?
Yeah, it's one of those sponges that is characteristic nodule fields.
Mm-hmm.
Sometimes we find them small. We found a few on rocks collected earlier in expedition, but no Asbestopluma yet, so this would be a new collection, new genus for the expedition if we can get that.
I have like our twofers.
You said that the scoop is in the bin because it was displaced during a morning attempt at collecting a 70-plus pound rock.
Nice.
I don't know if that's the right tool for the job.
Uh-huh.
Did they successfully collect that rock?
No. The rock had to be offloaded. It was too heavy.
I wonder who did that.
I believe it was Trevor.
Yeah, do you want to follow me in on that camera there, please?
I woke up to an email this morning from Lindsay saying, Lindsay G at OET, saying we might need to get a second pallet for that rock. Yeah. So it's probably a good thing that it was not.
I think Trevor was trying to sway the total weight.
Yeah.
How many rock samples have we collected on this expedition?
I think to go back and check. I don't know. I'm pretty close. I'm pretty sure it's close to infinity. infinity rocks.
Yeah.
Kidding. So in total, this will be our 172nd sample. And as of midpoint on the cruise, we had 350 pounds of rocks. So I would say we probably doubled that.
Yeah, but then we
Yeah.
we got really
Yeah.
into some big heavy rocks.
Yeah, so I think it's safe to say we're probably going to have close to 700 to 800 pounds of rocks on the ship by the time we reach port.
Put them up on a box here. There we go.
Come on, scoop.
Go ahead and close up that bin there, Jake. Go ahead and push on in there, please. That's great.
So let's reorient ourselves. Where would that?
The sponge is right between your toes. Yeah, it's a little close.
Oh, you want the one with the sponge?
It was like right in there, right?
Yeah, it's attached to the nodule, so it should come out.
Yeah.
Fairly
Just want to make
easily.
sure we get it in the scoop.
I'm going to reset my grip here a little bit.
Some of these nodules do have botryoidal textures, the larger ones. Oh, wow.
Yeah, I don't even want to think about the
Yeah,
shipping.
Kevin, I hope you guys have plenty of care for your backs on that cruise with 5,000 pounds of rocks. That might be, it might actually just be easier to grab that particular one.
It might be, yeah. Well, we can scoop others along with it. It doesn't just have to be that one.
I'm worried the sponge might get chopped off.
Okay, good point. So Jess, if we can maybe, yeah, exactly. Let's scoop over there, and then we'll come back and pick up the one with the sponge on it.
Sorry.
That looks great.
All right, Jake, you want to open up that bin again, please?
Sorry, what?
You can open up the bin, please.
Yep.
Whoa.
Can I check the DV losses there, Jake? On the fibers?
Yeah. It's on that engineering computer, yeah. I got it.
Okay. Don't want to do that.
Great. And then, Jess, if we could,
Go ahead and close
with
up
the manip,
box.
try to pick up that additional nodule with the sponge attached, and we can add it to the bin.
Sure.
And can we also, when we zoom in again, get the lasers on? I don't think we got a shot with that in.
Go ahead and push on in there, please.
Thanks, Jake.
That's great.
I think it might be just behind one of those toes.
Oh yeah. You want to look at it on the bubble cam there, please?
Yeah, there it is.
Full wide
It's
there,
that
please.
one right there.
Thank you. All right, go ahead and push back on in there. All right. It's about there.
Got it.
All right, Jake. Oh.
Yeah. The
That
rock.
might have just been it.
The rocks readjusted themselves.
Yeah.
We can wait a minute for the sediment to clear out. Maybe while we're looking, get a partial zoom on the rock that looks like it broke open.
Go ahead and push on there, please. Oh, it's still there.
Very gently cradled.
Full wide.
Thank you.
Jake, you want to open up that bin?
Yep.
And make sure I'm full rack back there.
You can turn the lasers off, Jake.
Great.
All right, go ahead and pop that in there, please.
That's 172.
172.
You can 2 on in there, Jake.
Asbestos
Yeah, I think
Sure.
it's Asbestopluma.
Sorry, Jess, what did you say?
Oh, we're just going to let Jake fly for the next couple of minutes of bottom time before
Okay.
we lift off.
That sounds great.
Beth, is it known what makes the sediment so light colored?
Well, it's has to do with the composition of the material. So some of this material might be broken up bits of coral, might be benthic forams, particulate matter from the surface ocean, all mixed together. Probably not a lot of organic material, so it doesn't have that darker brown color that you associate with muds that you see
All right, you're
in other continental shelf settings.
We'll just call in that one final ship move.
Yeah, let's do it.
Bridge nav.
Some interesting looking basalt textures
Step
here.
50 meters, bearing 155. Thank you.
Just a little heads up. Before we come too far off bottom, eventually, maybe 20 meters or so, we're going to do one final Niskin.
20 meters off bottom or 20 meters vertical? What do you?
20 meters off the bottom, so
Okay.
altitude, yeah, when we do our
Got it.
start our recovery mode.
Yeah, that'll be a good recovery moment once we come up in the water column.
Good flow here.
Yeah,
And
still some
east
nice coral
towards the-
thickets.
Yeah, it's east towards the west.
Yeah, exactly. East towards the west. But still, surprisingly, none of those, or at least not yet, big polyopagons up here. Although they were seen elsewhere on this seamount. Not up here. I was kind of thinking we'd find some.
I'm a little surprised to not see any sea pens up here, even though the sediment is not particularly deep.
Mm-hmm.
You know, you typically find sea pens at this depth, like maybe even a rock pen or so, but at least the Protoptylidae, Halipterus sometimes, maybe a little bit too deep for Halipterus, but this would be like typical type of substrate where you'd find them, where you have this patchiness.
Yeah, these little pockets of sediment.
Steve, the coral with the spiral stalks are Ridogorgia?
Yes, yeah, we've had at least 2 species in this site. I think a bit deeper on our watch last night, we were seeing Magnospiralis and Iridogorgia bella are these more tightly whorled species.
So we're coming up on our last bit of time here on the sea floor of O'Oikaika Seamount. In fact, our last moments on the seafloor for this expedition.
There's another one of those candelabra type corals coming into view. Some It's nice just to sit here and soak in the view as we
You're getting tugged.
come into these last moments on the seafloor. Still quite a bit of sediment on this side. Haven't quite come around to the eastern side where the scouring might be more pronounced.
Hey, Steve. So for the rock samples that have been collected, about how, do they know about how large they are?
Steve, did you hear that?
Sorry, can you repeat that?
I think it was Jess.
Yeah, for the rock samples that have already been collected,
Yes.
do they know as to how large they've been?
We're trying to figure out how much weight to drop.
Yeah, I've only seen, I don't know how big they are, but I've only seen the box when it was open, the starboard box, and some of them on the outboard side are quite large, big boulders.
Okay.
I would say heavy.
Roger. Thank you.
Seas are starting to pick up there. Doesn't feel so bad in the boat, but when you start to look aft at the seas and imagine an ROV bobbing around up there.
Yeah, swinging around.
Trying to get it back onto a stationary position can be.
Yeah, we'll want to be really extra cautious. Yeah, definitely feeling that roll up here.
Yeah.
For those who aren't familiar, the control vans for ROV Herc and ROV Argus are up towards the top of the ship. So we definitely feel an extended roll whenever the ship is moving.
It's one of those yellow ones up there.
Oh yeah, way up in the top right corner.
It's Escalade.
Where is that? Yeah.
I don't know that for sure.
Dead center, top of the screen, 12 o'clock. Is that what you were talking about?
Yeah.
Yeah. Could be. There, yeah, it might be worthwhile to take a look at some of these other bamboos that have the yellow wash on the base.
Like this one right in front of us?
Yeah, I'm thinking about the one in the background, actually. It looks a little bit less S1-y. Top of the screen, 12 o'clock.
This one up here.
Yeah.
Did you assign it an S number yet, Rennie?
I might have to retract that based on new information. AKA what Steve says it is. That's the new information.
I'm just relaying what our scientists ashore are saying.
And they're just making it up, so it kind of just comes full circle. Oh, wow, this is
really yellow.
Spectacular.
This one is, yeah, it is odd because it seems like it has greater propensity to branch the further out you go on the branch tips, so more distal.
You want to zoom in on anything in particular, Steve?
Just everything.
Okay.
Yeah, kind of base to the top we can.
And can we get the lasers off, please?
Yep.
Reminds me of the tree in Go Dog Go. It's got all the little brittle stars on it.
Because it's got some weird, interesting morphology on the bottom, too. Thanks for the sound effects.
Jeff's narration is always very entertaining.
Rennie hit it earlier yesterday. It's a Wilhelm scream.
Yeah.
It gives me an idea of stitching together a Nautilus Live action movie.
Seems that all these polyps are kind of oriented down. Is that?
Yeah, that's. I don't think we saw that on the previous ones, or at least it wasn't as apparent.
Are the ones at the end a lighter color, or is that just because they're closer to the light?
Yeah, I do think they are. Yeah.
It looks like they're lighter.
Yeah,
And a richer yellow and lower.
I'm always confused by these yellow escalades because sometimes it's only the bottom few inches of the base that is yellow, and then other times it's the whole colony. Yeah.
The polyps also seem like featherier. How do you say that word? More feather-like.
Yeah.
Can we go down towards the bottom when we have a second? Yeah, it looks like nodal, well, just above the node branching.
Yeah, that does seem to be just above the node.
It's tough to tell. The deeper you go into the colony, the more calcified over the nodes become, and they become a little bit more difficult to discern. Well, that one's doing a trick. Look at that.
Yeah, the brittle star is just
One
hanging
arm.
on.
Acrobatics.
This is my shot. Camera's on. Really large there at the base.
Yeah.
Yeah, that
Very
color is
yellow.
striking.
I really want that brittle star to photobomb us right here.
Yeah.
It's almost like roots at the base.
Yep.
Stabilizing.
Yeah, maybe failed branches or broken branches that have been cauterized by tissue.
Definitely seems to be an increased current here from east to west.
Okay.
Just based on the position of Argus relative to the ship where,
Should then be pushed back.
yeah, it's
Yeah.
quite
I'm
laid back. And
happy with this.
That's
Oh.
great.
All right.
Thanks, Jake. That was a
Yeah. Very nice.
lovely colony to spend some time on in our last few moments here on the seafloor.
Yeah, we might
Yeah.
want to start to set up to drop the weights and then go up and pop the Niskin.
Okay. Yeah, let's look for an area that's relatively low density for.
So I'll sit down and then we'll switch back.
Yeah.
Try
What's
to-
going to be your recovery heading there, Rennie? Or ish?
Ish, east.
East, Raj.
So you'll go west. Yeah, try to find a patch without, with less in it, or yeah, maybe over there.
Maybe on
drop.
the right. Yeah, it looks like there's some bare spots that we
Yeah,
can
over here.
dispose of the dive weights.
Nice. That's a good spot.
And a dead Walteria sponge on the way out. So there is a sponge up here. A single sponge.
Single sponge. All right, I'm going to face the recovery heading with Argus here.
Okay.
It looks like box E has the biggest rock. It says it's about 20 centimeters diameter, but
Nice.
from my experience, just double that.
Yeah.
All right. Full rack back. You want to grab the weight? I'll get the Niskin.
Sure. Are we dropping just one?
Yeah, I think so. Maybe try the one over by Mungo since that one's unsecured. Follow you in on bubble here. If that line's too hard to get over, then we can get the other one. We'll just stow Mungo on top of it. So it's one of the two. Nice.
Jeff, do you mind putting in the over the pilot view so folks at home can watch how this manipulation is done?
Gross. Yeah. Nice. Very nice.
So for any viewers at home that have never seen this happen before, what we're doing is basically trimming our ballast a little bit. We picked up some rocks on this dive, so we need to get rid of a little bit of excess weight. So we're remain positively buoyant. This is a steel plate with a natural fiber rope attached to it. Both of these will degrade relatively quickly as to not leave a lasting impact here.
Nicely done.
All part of our preparations for ascension.
All right. Switching back.
Thanks, Jake. Do we want to wait to do our release ceremony till we've gotten the water sample?
Yeah, 15 to 20 meters off bottom, something like that would be ideal.
Yeah, we'll get that there, and then we'll start our ascent, and then that way we can do it while we're in motion.
Yeah, okay. Seeing our last views of the seafloor here of this expedition. It's been a really interesting dive. Different features that we've seen as we've ascended about 350 meters or so throughout this dive from the northwest up to the summit here. Definitely some strong currents coming from different directions depending on which part of the seamount we're on, although that might also be related to other factors as well. But
You want to come up a little there?
seems like a well-fed community here for both the corals and the predators. The final sample that we will collect will be a water sample, a bit off bottom, that we'll use for an eDNA study that is part of this expedition. The concept here is that animals in the water shed cellular material that contains their DNA, and we'll capture that on a filter and use it for identifying the organisms without having to sample them. By getting one a bit off bottom, we can use it as kind of a calibration for the samples that we collect much closer to the seafloor and much closer to the animals to kind of see how far that signal travels.
Yep.
You can definitely see that there's still a lot of particles in the water in the HertCam. So those will be the material that we capture in our Niskin bottle.
All right, Steve, we're at 23 meters off bottom here.
All right.
And it's the last Niskin that needs to be
6.
fired. 6, Raj.
Jeff, can we see the Niskins in channel 3, please? Right, so you can see that final bottle that is left to be closed on the very bottom of the frame. So as Jess pulls on this lanyard, it'll trigger the top and bottom of that bottle to close, snap shut, collect the water that's on the inside.
Jake, you want to watch my altitude there?
It's because we're going down slope.
Reg.
Bottle's closed.
All right. Great. So what was the final altitude on that?
It's about 30 probably.
Yeah,
Usually it resets at 30.
just about 30.
And what's our final sample number?
173.
173.
I'm going to switch you over to USBL.
Not bad for 13 dives.
Yeah. That's right. That's in the range. You know, a typical 2-3 week cruise is 150 to 200 samples.
Mm-hmm.
About that.
All right, front row, just let us know when you're ready.
Okay, we're going to get set up and start our ascent, and then we'll let you know. We'll be good to go.
All right. About 15 going up there.
Anybody have a pen handy? Steve, may I borrow your pen? Thank you.
Stills cam is not shooting intervals now.
Are we doing pretty steady?
Yeah, just about. All right. Yeah, go ahead, back row. We are ready.
Do we want the lasers on for our blue water ascent?
Sure thing. Okay, we're ready for the release whenever you guys are.
Okay, Lindsay, can you get us into this space?
Sure, yeah. So for our final time here, if you've been following along on this expedition and the end of our dives, we take a moment here to say a thank you. It's a personal and collective thank you to the space, to this region. We're also going to be taking this opportunity to mentally break away from the endeavor that we've had and move on to our next tasks. So we're going to go ahead and be silent. I'm going to invite folks in the control van to go ahead and mute your microphones, and we'll be back with everyone around the world shortly.
For anyone interested in learning more about the cultural protocols and connection to Olelo, Hawaii for this expedition, I invite you to go to the nautilusdive.org website. And when you see that expedition status, you see the name of our expedition. If you click on that, one of the first blog posts associated with this expedition talks about that approach, and you can see some of the naming and thoughtfulness that went into how we want to approach our work here. Also, if you go to the Office of Hawaiian Affairs.gov webpage and look for the Papahānaumoku Kuakea part of OHA.gov, there's also some information there about some of these protocols. So you can follow along and learn more about how we've been trying to center our work here in this space that's considered sacred by the Native Hawaiian culture. And I'd like to thank the Native Hawaiian Cultural Working Group with the Office of Hawaiian Affairs and co-trustees of the monument for engaging with Ocean Exploration Trust and our scientific team as a partner in this work and helping us develop these protocols and names and story that'll be part of this broader expedition series. We've got about 1,500 meters of ascent through the blue water above Ho'oikaika Seamount. Still lots of particles swirling about, lots of microscopic life in front of us to admire as we come up to the surface. Should be on deck around noon Hawaii time. And then we'll begin our journey back towards Oahu. It'll take us a few days. The weather's been picking up, the swell's been picking up, and it's going to be hitting us on the beam as we go back. So we'll be rolling a bit on our way home and leaving a little early so that we have plenty of time to get in and get ahead of this storm that's coming up from our west. We will be multi-beam mapping as we continue back. So you are welcome to stay tuned in to our live broadcast and watch the multi-beam data coming in and watch the ship as it's transiting through the monument and back into Hawaiian waters. We won't be monitoring the chat though, I don't believe, as we're doing the multi-beam monitoring. So if you would like to stay engaged with the Nautilus team as we begin underway, please use our social media channels. There'll still be posts going up, some content from this cruise being posted on the webpage, some of the highlights and things of that nature. Anybody want to share any thoughts as we're coming up about what you think of that last dive or this expedition in general?
I haven't seen such large sponges before like that. Those are large satellite dish sponges. I've never seen those before. That was very exciting to fly around.
Yeah, definitely some cool sponges out in this region.
Yeah, I've been collecting some nice words from our viewers. I'll finishing up our status update, but I will read those in a moment. They're really nice.
Can I ask the front row what's going on with the Argus cam?
Oh, we had a hard ground fault on Argus's mini Zeus.
Okay.
That's been on kind of the whole dive.
Yeah.
But to save whatever's, to kind of give it a rest, if you will.
Okay.
We switch it to the dash cam so you get the 90s kickback, throwback right now.
Great. So then our audience might want to just stick with channel 1 for watching the blue water go by.
We could put like the Malcolm in the Middle subtitles up there next, and then start shaking the camera around.
It's okay. As a reminder to our audience, the samples that were collected on this expedition, the majority of them are going to be going to archives so that scientists from around the world can request them to work on. The animal collections will be going to the Museum of Comparative Zoology at Harvard University. And the rock samples for geological analysis will be going to a repository at the University of Rhode Island Graduate School of Oceanography. The water samples that have been collected for the eDNA analysis go to a lab from some NOAA researchers.
Yep,
to coordinate that work.
Northwest Fisheries Science Center.
Thank you.
Right now there's, since eDNA has been kind of something that we've been working on developing methods for sampling for a while now, there is no repository that we've really established for those collections, so they're going to a partner lab, Meredith Everett's lab at NOAA, for storage and sequencing. She was also the recipient of the previous monument cruise collections, eDNA collections in 2018. Yep.
The NA101.
Yeah, so we have a very good, now we have data points from north of the, the northern parts of the monument as well now as the southern parts of the monument.
Yeah.
As well as throughout the Pacific remote islands and throughout the Pacific remote islands down as far as Phoenix Islands.
Will there also be samples collected on the upcoming expedition to the Chautauqua seamounts?
Yes, yeah, we've saved a rack of tubes. Usually we're limited by just the amount of preservative we have on board and the number of filters we have.
Yeah.
And less so by the Niskin bottles, because we can always take kind of pseudo-replicate samples of the material that's in the Niskins, and sometimes we do that just to evaluate how consistent eDNA is dispersed in a Niskin water sample.
Yeah.
But yeah, we'll have about 25 or 30 samples to take on the next cruise.
Yeah, so again, for our audience, although we're heading into port, shortly thereafter, another expedition will start, visiting some other seamounts around the Hawaiian Islands. I don't believe those are in the monument. They're outside of the monument boundary, but still, again, adding to our greater understanding of seamount ecosystems in this general part of the world. And then in early 2022, sometime in the April timeframe, another Ocean Exploration Trust expedition will take place at the Liliuokalani Ridge, which is pretty much due north of us on the other side of the Northwest Hawaiian Island chain. Those seamounts were mapped on an expedition of Nautilus that happened just a month ago or so. So the exploration will continue.
As drawn, the Chautauqua Seamounts range includes one seamount in the monument.
Okay.
Or partly in the monument. The summit is at least. And so that it's up for debate whether we're going to visit that site or not.
Okay.
But it's on the schedule as a potential dive site.
And even though it won't necessarily be in the monument, I believe it's still part of the expedition story in terms of having a Olelo Hawaii name.
Yes.
And trying to incorporate some of the cultural practices that we have been exploring together on this expedition. Rennie, I see that you're pulling up our weather forecast. Can we maybe show the audience in Channel 3 what we're facing in our immediate future?
Sure.
What we're up against?
What we're up against, yeah.
Which PC?
Oh, this is just HIPAC survey, HIPAC 2.
So, yeah, there we are, our current position and the weather front that's
Yeah.
quickly approaching us.
This is the main Hawaiian Islands where we'll be heading. That's about from where we are to there to Honolulu is about a 3-day steam. And we got this system here. This is actually waves. By the way, this is just windy.com, so
yeah,
anyone can pull this up. We have a system coming through. So let's see, it's 24 hours from now. You can see the system starts coming towards us. those are the waves.
Yeah, hopefully we'll be far away from there.
24 hours after that. Yeah.
Yeah.
So we won't quite, we'll be in there.
Yeah.
On the transit.
Bad.
Red means bad. And so,
it's going to be a bumpy ride home.
Yeah, that'll be towards, let's see, I'm just going to go ahead and guess. So that's one.
Hopefully everybody saves some of their seasickness medicine.
So, yeah, we'll be right on the cusp of it. Those waves are 4 meters with 2 meter swell, so that's 6 meters, 7 meter seas, plus 35 knot winds. So yeah. On Friday, we'll be having a good time.
Definitely want to tune into the Nautilus feed those days and so watch how the ship is bobbing around.
Friday may not be the best day to do laundry.
Yeah.
Oh my goodness.
Definitely going to see some white water over the bow.
Yeah, and so I'm sure the captain will be figuring out the best heading if we have to zigzag, because we do have a little bit of time that we bought by leaving early, I think. So we may have to zigzag in order to save our lunch. And eventually we'll end up closer to the main Hawaiian Islands. Let's see what it looks like. This whole system is coming towards, so it will be following us.
Yeah, it's going to be a rainy D mode.
Yeah. So, okay, so that was the waves we were looking at.
And yeah, that's just the waves.
Let's take a look at
Is there rain associated with this, or is it just windstorm?
Let's find out. It's gonna... is this colored
I bet this probably means there's probably some decent surf conditions on the islands.
I would imagine so. Definitely on the North Shore.
Is that something, folks, is there like a threshold where it's like unsafe to go out?
Depends on probably your skill level.
Thinking
and
about
insanity.
Not sure if they close beaches or something, or is that, yeah.
Yeah, there'll be some rain.
Okay.
The other issue with like doing interactions on Friday when we're taking seas like that is the dish can only keep up so far.
Yeah, right.
Yeah.
It can only move so fast. So if anybody's ever seen a picture of the dish that's in the back of the boat here, it's basically a satellite dish on roller skates. And it can move. It actually, if you look at the wheels, they really do look like roller skate wheels. And it can move really fast. And it's very finely balanced, so it just takes a fingertip and the thing will move. But at some point it says, no, I can't keep up.
Right.
And so as we start to pitch more and roll more, it eventually says, I'm sorry, I'm done. Which is really bad because everybody's hunkered down in their cabins trying to, read or whatever on the internet, and the internet just keeps getting slower. So.
I don't know that I will be trying to read if we're pitching around.
Well, I always wondered about that. The ship's always moving, the satellite's moving. I mean, what's the lifespan of one of those motors that controls the satellite orientation?
We replaced one earlier. I don't remember if we replaced a motor or one of the amplifiers earlier this year. But we did replace something earlier this year.
I think it was the amplifier. Was
it the amplifier?
Just half remembering ship rumblings from shakedown.
I just remember a couple of guys running around.
It's a long time ago.
Grabbing a ladder and all that.
Been a lot of crews that's happened.
It's all starting to blend.
Yeah. That was back in June.
Jake and Jess, will we be doing anything special to secure Herc and Argus with the coming conditions?
We'll put it into the hangar, so we'll bring the vehicles in, or at least Herc. Can't do anything with our guys back there besides secure it down, cover it with tarps, and yep. So we have ratchet straps on all the vehicles that we strap it down to the deck. And then, yeah, pretty much it. Make sure things don't fall around in the shop.
Yeah.
So just generally securing things for sea.
Secure the cribbage board.
Yeah, I
That'll
think
be
after this
secured.
watch, we'll all be running around trying to make sure everything's secured.
Yeah.
And see what kind of mapping we get on the way out. But weather may prevent that, and also the way we have to travel may prevent it from being useful. So
I'm going to try and get the rocks dried out, but I don't know how we're going to dry them out and secure them at the same time, probably on the floor, boxes, stacked together. Don't go in the wet lab for your life.
Yeah, unless you want to break your ankle.
yeah,
Might be some kneecappers rolling around in there for a little while.
I'm going to set up the tape with start and finish. And we have to name the boxes after racehorses.
Come on, box 20. The utility room has been invaluable. Full of rocks right now.
What was that word? Oh, lateralia. My racehorse is lateralia.
Yeah, that's what we had to move out of the way the other day.
Yeah. We'll move those out of there once we reach port. Have some more permanent storage.
Well, we had to get something out of the utility room yesterday, that was in the absolute worst place that we could get to.
Yeah,
move all of these boxes, move the UPS, move the monitor, unstrap this.
I walked in on Jeff and he was boxed in back there. It's like the opposite of the room of requirement. It's the futility.
Yeah,
futility room.
It's the futility room.
General purpose dumping ground.
Yeah.
Yeah, in a former life, it was the studio.
Right.
Yeah.
And in the
That's
former
right,
life, it was a sat cave.
Yeah.
You ready to hear some kind words from our viewers?
Sure.
Yeah, sure. Why not? Let's do it.
All right. There's a lot. I'll just read a few.
Thanks to folks who are tuning in.
Yeah. So again, this is our final ascent on this expedition here. So in the UK, we have viewers who say, you have been such a calming and soothing staple in the household for the length of the entire expedition. Not just the beautiful biology you've been finding, but also the perfectly done commentary during every watch. You all get on so well, have a great sense of humor, and are so passionate about what you do. It's been an absolute pleasure to listen to you all. I don't know what we're going to do with our evenings after today.
Nice one.
Well,
Thank
thank
you.
you so much. Please stick around with Nautilus because there's going to be more expeditions coming up.
Yeah, we can't promise our good humor, but...
I'll work on some fresh material.
Yeah. Well,
Actually, yeah, a lot of this watch is changing out because Steve's staying on, but he's changing watches